Anyway, all right, we can just start, and I'll let you know when to switch the slides. Well, first, I'd like to start by thanking Brian Johnson for his talk. I think his mission and vision are truly inspiring, and I'd like to thank the Wonderland team here from Microdose for having me out to speak with you today. I'm really excited to tell you about an exciting breakthrough we have at the intersection of psychedelics and longevity. But first, let me tell you a little bit about myself. So I'm Sam. I did my undergrad degree in neuroscience at MIT, and my MD and my PhD at Columbia. I, like many of you, have friends and family members who struggled with serious mental illnesses and diseases of neurodegeneration. And during my medical school years, I came to realize that the current therapies are woefully inadequate, and there's been a lack of new therapies for mental illness over this past decade, and almost nothing for healthy aging. So in 2017, I left my career in medicine and academia to found a biotech company, Terran Biosciences, to help fix this. Now at Terran, we take a unique approach to drug development. Unlike the traditional approach, which uses a hierarchical top-down management, we have a very small team of highly capable operators who then work to build out individual teams of world experts to solve specific problems. At Terran, I assembled some of the brightest minds in the industry across drug development, discovery, medicinal chemistry, clinical practice. And with this approach, we've had a lot of success. The first was bringing back a therapeutic from the 1990s. This was the first adjunctive treatment for schizophrenia. When added to a patient's baseline antipsychotic regimen, it was able to double the efficacy above the baseline treatment. Despite all the success in clinical trials and a good safety profile, it had a drawback, which was it had to be given 3 times a day. And so the drug was deemed unfixable, and development was halted. We spoke with the doctor who ran those clinical studies, and he said for years afterwards, patients and their families were calling, begging to be put back on the drug, but there was nothing they can do. Manufacturing had stopped. So once we learned about it, we flew to France, met with the big pharma company, acquired the rights to the drug, built out a team of world experts, created a once-daily form of the drug, and put it back in the clinic. Patients have waited 30 years for access to this drug because it's the only drug in its class with this mechanism. And we hope, if approved, this could be first in class, not only for schizophrenia, but also for neurodegenerative disorders where it's also shown clinical success previously. Now, around the same time, we did something else, also in the neuroregeneration and aging space, and that is we came across a biomarker for aging called neuromelanin and for neurodegenerative disorders. Now, this was a biomarker that you can measure on MRI in 5 minutes and had been used clinically for more than 20 years in hundreds of patients. But that was only in the research setting. It could never be FDA approved because despite doctors calling for it to become part of the new standard of care, saying everybody should get one after age 40 every decade to measure brain health, there was no way to standardize the measurement until Columbia University built a team around this, and they found a way to create a new algorithm to fully standardize the measurement of the biomarker. Then we licensed that technology, built out a team around it, and then built a fully cloud deployable automated MRI analysis system that integrates directly with hospitals. And in a big win for patients, we just got FDA clearance to launch, the first FDA clearance of this biomarker in history. Thank you. where we hope this will help revolutionize the treatment of neurodegenerative disorders, early detection, and also contribute to healthy aging as well. So at Terran, we're a platform company in this regard, where we take this approach in order to new and exciting opportunities with the goal of bringing promising treatments to patients in need. Now, we'd also been following the psychedelic landscape very closely for a number of years, and we were impressed by their ability to not just act as therapeutics, in fact, they act as rapid antidepressants, but also to be able to be neuroregenerative. They had the capability to cause adult neurogenesis, where you can regrow neurons in the adult brain, a capability only thought to occur in infants and childhood. However, it wasn't until a really exciting breakthrough we came across in the academic space that we found to be absolutely game-changing that we then entered the psychedelic space. Maybe we could advance to the next slide. So this was the work of Dr. Jordi Riba from Spain, one of the pioneers of understanding the mechanism of ayahuasca therapy. And what he showed, and this has been replicated by other labs, is that DMT induces neurogenesis. If you could advance the slide, please. Now, that led to the question the researchers asked. They said, well, we know psychedelics act on a lot of receptors in the brain. And we also know that only one receptor is responsible for the trip, the serotonin 2A receptor. If you could advance the slide, please. So as you see in the plethora of, oh no, can you go back one slide? That'd be great. Right there, that's perfect. So, oh, it's working now. Thank you. Okay, so we know only one receptor is responsible for the trip. So they asked the question, what would happen if we took a psychedelic that's already effective, causes neurogenesis, works as an antidepressant, and we combine it with a drug that selectively only blocks the serotonin 2A receptor? Would we maintain the benefits of the psychedelic without the trip? Now let's just break this down for a second. We're not anti-trip. We know the trip can be incredibly valuable for patients, and in some cases, like post-traumatic stress disorder linked to a traumatic life event, may be absolutely essential. But in terms of disease like depression or neuroregeneration, healthy aging, we know that rolling out a psychedelic with a full multi-hour trip, where the FDA has said you have to give it in a hospital setting or a therapeutic setting, presents a number of barriers to patient access, including the time it takes, financial access, the doctor's time. And so if there were a way that you could have a take-home medication for those conditions, you could potentially reach millions more people with the benefits of the psychedelic. So they asked that question. They redid the experiment, and they found that when you combine DMT with a selective serotonin 2A blocker in the Petri dish, in vitro, you maintained all the same benefits. In fact, on measures such as neural growth, you get the same effect, with and without the serotonin 2A receptor blocked. And we know in humans, if you block the serotonin 2A receptor, you remove the trip. And they had other measures of the diameter of the growth, and similarly, blocking did not remove any of the beneficial effects. And these have been collaborated at a number of academic centers, and we'll just go through a number of the studies here using DMT, psilocybin, LSD, and a number of other compounds. And there's been a number of explanations for this mechanism. But at Terran, we wanted to make sure that this actually translates to humans, because we know that scientists have already cured all diseases known to mouse kind, but it doesn't always translate to humans. And so we decided to do the data analysis study with our team we're collaborating with at Yale on an imaging study to understand if the effects of the neuronal effects on rewiring and functional connectivity induced by psychedelics in the brain would be conserved. Now, this was a study that was done in Europe where they had given 2 placebo pills, given MRI scans, and then measured if there was a trip. It's a crossover study, so everybody gets every condition. So after they get that, then they get 1 placebo pill and a pill of LSD. I believe 100 micrograms. So they get the same scans to measure the functional rewiring of the brain on LSD, and they're experiencing a psychedelic trip. And then, a few weeks later, they come back, and they get, we lost connectivity. Oh, oops, there we go. It works. And they get 1 serotonin 2-way blocker pill and LSD. They get the same scans, and they have no trip. So you take these and you subtract the baseline condition so you understand the changes that are occurring when you have the LSD alone, and you have the changes that are occurring with the block version where there's no trip. Now, what we found was really shocking. On both metrics of functional connectivity, the functional network assignment, FC matrix, and global brain connectivity, we see the same pattern with a high correlation of rewiring of the brain that occurs on LSD, and this is similar to what's been shown in other studies, also occurs on the blocked version. The big difference being that people here are having an intense trip, and the people here are not. And the map of changes in functional network assignment in the brain that you see on the left, that's the map where there's no trip. Those are the changes, because it's subtracted from placebo, that LSD is inducing without any trip. And so we'll take a quick look at the detailed data. This shows essentially the scans through all the subbrain regions and the basal ganglia. At the top, again, is LSD alone, and at the bottom is the block version. And as you see, the LSD alone is a little bit more intense, which is to be expected, because with the block version, we have access now to do things not done before, where you can take multiple pills, you could take pills multiple days in a row, and thus, can we move the window off the side there? Thank you. And thus, potentially achieve the same effect. So then, okay. We lost the clicker. Can we advance? Yes. Can we go back one? Advance one slide? No, we. Okay. So just to show the math behind it, and this is that essentially the LSD versus the block version correlate together with an R-score of 0.8, which is extremely high, showing the direct correlation of the changes that are occurring without the trip. And then on functional connectivity, the changes induced by the block version versus LSD alone, an R-score of 0.64, and we're seeing that essentially LSD is causing network changes in the absence of hallucinations, consistent with the plethora of preclinical studies published in this regard as well. So then what we did was we licensed the technology from the university. We went to Sanofi, who had the world's most selective and safe blockers, epilevancerin and volaneancerin, and those had been tested in over 15,000 patients. We licensed those on as well, built a team of experts, and now we're moving this compound forward. So I'm going to show you a quick video that explains exactly what we're doing with this partnership. Okay, we got to go back one. Okay. There we go. Do we have audio? Maybe not. Thank you.
However,
Oh, there.
psychedelic therapy often requires a supervised multi-hour psychedelic trip and comes with the risk of a bad trip, and thus may only reach a very limited number of patients as second or third line therapy. Terran is developing therapies combining psychedelics with serotonin receptor blockers that may have the potential to deliver the therapeutic benefits of psychedelic medicines without the associated hallucinogenic trip. This approach could increase the potential for broader clinical use of psychedelics and could expand access for many patients, including those with major depressive disorder, anxiety, and neurodegenerative diseases. Terran's non-hallucinogenic psychedelic therapeutics may have the potential to become first-line take-home therapies with superior safety, efficacy, and more rapid onset compared to standard antidepressants. In parallel, Terran is developing new improved forms of classic psychedelics for other indications where the trip is thought to be an essential part of the therapeutic benefit, such as PTSD. Additionally, Terran is developing receptor-blocking compounds that may be able to end psychedelic experiences or tailor them to a specific duration. These compounds have previously been tested in over 15,000 patients across a variety of indications. Terran has formed partnerships with universities and clinics and is providing psychedelic compounds to researchers globally. With our unique approach, Terran Biosciences is well-positioned to transform the treatment paradigm for patients suffering from neuropsychiatric illnesses.
All right. We didn't just stop there. We then went on to make a number of other breakthroughs with different teams assigned to that, including the world's first long-acting MDMA, oral prodrugs of DMT that are orally active without the MAO inhibitor, new forms of psilocin, new improved versions of LSD and 2CB, and new versions of empathogens. And now with this approach, we have three things we're working on. One is the blocked take-home compound that could be useful for neuroregeneration, healthy aging, rapid antidepressant effect, all possible future uses. And then we have psychedelics with the trip for the conditions where you might need a trip. And then we have the blocking compound alone, because by taking that, you can end a trip at any time point and thus tailor a trip to a specific duration, which can now make therapy more manageable. For example, LSD with 12 hours might not work in a therapeutic session, but if you could tailor an LSD therapy session to maybe 1 or 2 hours, that could be much more useful in a hospital setting. So I'm very proud of the accomplishments of our team, but the work's not done yet. Mental illnesses are very variable, and we need therapies, as you've seen here, that are as diverse as the illnesses they treat, so that we can offer right-sized solutions for patients. We're not content to just sit idly by and hope somebody else will do it, so we're going to continue to build out the Terran engine and the platform with new compounds so that we can keep bringing new therapies to patients. If you'd like to learn more about what we're doing, we have a workshop tomorrow at 2 p.m. Titled let me think about Finding the Path, the Intersection of Biopharma, Longevity, and Psychedelics. Thank you.
Do you want to take some questions?
Sure, I want to share how much time there is, because that's always been on zero.
Oh, no, you got a couple minutes.
Okay.
If you,
yeah,
we have
a
time for a
questions.
couple questions, if that's cool.
Sure. That's right. I think, and we've seen that.
Hold on. Could you actually repeat the question in the
Oh,
microphone?
yes,
so that
that's
other
a good
people-
point. So the question is that by blocking 2A, we're still maintaining the cascade of effects from other receptors, the psychoplastogen effect, and growth factors. And we would agree with that. There was actually a recent paper published that used volaneancerin, one of the compounds that we've licensed clinically from Sanofi. But they used volaneancerin to block the 2A, and they said, well, you know, LSD is exerting, and psilocybin, there's psychoplastogen effects through TrkB receptor, BDNF binding. There's been other papers saying maybe it's other receptors, but in general, the general consensus we believe in preclinical literature is you don't need the 2A receptor to have all these beneficial effects. Yes. Yes. Oh.
Question back here.
Oh, okay. Yep.
First of all, thanks for being here and congratulations on all the amazing work. It's really fantastic what you guys are doing.
Thank you.
So I just had a quick question about the scientific development process that you guys are going through. So I did some banking work for a company called Therapeutics, who was trying to do the same thing basically with ketamine, which was ketamine without the trip. And they got into some complicated issues around clinical trial design, going through the regulatory process, trying to prove the relevant endpoints and data markers. Can you talk a little bit about how it's different with these compounds and different now with like a more supportive FDA and how you guys are kind of like walking through that process?
Yeah, for sure. So with ketamine, it is a little bit different because there you have a compound that's already kind of has a wide rollout already, and it's a dissociative anesthetic and a little bit of a different mechanism, you know, mainly thought to occur through the NMDA receptor, but also involves opioid receptors and several others. And the idea is with psychedelics, it's a little different because there you have a highly selective effect, a mechanism that explains the trip, the serotonin 2A receptor. And there's already been studies in humans removing the trip where by just blocking the 2A receptor with ketanserin or other antagonists. So for us, what we're trying to do is just develop this into a pill where we would show in clinical studies that there is no trip at all, and then move that forward through the traditional regulatory path with the FDA. And now with the new FDA guidance, you know, the FDA has been very receptive to making it clear what they want from psychedelic companies. And I'll say one last thing too. This, we believe, falls under the regulatory path of one active compound combined with a second compound that reduces side effects of the first compound. And when you have that, then the FDA in the past has not required multi-arm trials where you have to show efficacy of the individuals. You just use the one combined with the side effect reducer. It's the type 2 path with FDA. But again, we cannot say for sure until we have more discussions with FDA what exactly, but those are our initial thoughts about some of the potential interactions.
So we have time for one more question.
Right, and did you have a question? Mm-hmm. We believe it is applicable. Yeah, sorry. Said, can we generalize from LSD to psilocybin and other psychedelics? And we think yes, because in the preclinical studies, we've seen this occur with LSD, with psilocybin, with DMT. Now, there may be important nuances in how you block the trip and the timing of how you block it and how you administer the blocker first at a certain time point before the psychedelic or after. And there's definitely some nuances there, but the final end goal of achieving a take-home pill, we believe, can be generalized to other psychedelics in the final state. All right, well, thank you.